Bismillahirrahmanirrahim. My dear students of second year MBBS, let's talk about the pancreas today. The pancreas is a compound tubulo-alveolar gland of purely serous type of variety. Rather, it is a mixed gland. It is endocrine as well as it is an exocrine gland. You are familiar with the islets of Langerhans, in which there are alpha cells and the beta cells and the gamma cells, and they produce insulin and glucagon. And these hormones, they control the blood glucose level in the body. As regards the exocrine portion of the pancreas, it is a serous type of gland. It secretes the enzymes which are poured at the ampulla of water in the second part of the duodenum and through the duct system. So this gland is exocrine as well as it is an exocrine type of gland. The stroma of the pancreas consists of a very thin connective tissue capsule. Pancreas, the exocrine gland is 97% and 3% is only the endocrine gland which secretes insulin and glucagon. When we cut a section, you will find that it consists of the parenchyma and the stroma and the duct system, just like the parotid gland. We can confuse the pancreas under the microscope with the parotid gland, but the only difference or the differentiation and the identification point will be the presence of the islets of Langerhans. So we will find the areas jazire honge jo ke they are surrounded by the reticular tissue, and these islets are having different types of cells. I will narrate later on. Let's talk about the stroma of the pancreas. It is covered by a connective tissue capsule which is thin, from which septa extend into the gland substance, dividing it into lobules, and within the lobules there are fine delicate connective tissue. The duct system comprises of the intralobular ducts, and the main excretory duct. The exocrine portion of the pancreas is a compound tubulo-alveolar gland. It has many small lobules, each of which is surrounded by the connective tissue septa, through which run the blood vessels, nerves and lymphatics, and interlobular ducts. Exocrine secretion by the pancreas is controlled by the hormones and the nerves. There are no striated ducts in the case of the pancreas. A unique feature of the intralobular ducts of the pancreas is that these ducts begin within the acini. So those ducts cells which are located inside the acinus are called centroacinar cells. The interlobular ducts lie within the septa and are lined by the simple columnar epithelium in which goblet cells are present. The interlobular ducts unite to form the main duct which is lined by stratified columnar epithelium. The parenchyma of the pancreas consists of both exocrine as well as endocrine components. The exocrine portion contains pancreatic acini.
The pancreas is composed of acinar cells, which comprise the exocrine pancreas. The endocrine functioning islets of Langerhans confirm that we are looking at the pancreas and not the parotid, which lacks endocrine function. First, let's examine the histology of the exocrine pancreas. These triangular or pyramidal secretory cells are called acini and comprise more than 80% of the adult pancreas. The nuclei are basally located and the cytoplasm is very granular. The granules differ between the apical and basal cytoplasms, and the granularity is the result of a high density of organelles. Here is a compact islet of Langerhans with well-defined borders and coarse speckled chromatin. Compact islets make up the majority of the endocrine pancreas. A small fraction of the total islet cells are more diffusely spread through the acini. In neonates, islets of Langerhans comprise about 20% of the pancreas, but only 1 to 2% of the total organ in adults. There are 4 types of endocrine cells. beta cells, alpha cells, delta cells, and PP cells. Each is present in varying concentrations and with distinct functionality, but it is not possible to discern the differences between the cells at this level of microscopic staining. Beta cells tend to be located centrally in the islets and may have larger nuclei. PP cells are very rare and make up the majority of diffuse islet cells. The pancreas contains a large amount of connective tissue. The septi divide lobes of the pancreas. We see arteries, veins, and capillaries throughout the connective tissue. Here is an artery and vein, and more connective tissue in the center of the image. In the lower right of the frame, we see our first large pancreatic duct. The pancreatic ducts are characterized by columnar mucinous epithelium. They have round nuclei which are located on the basal layer. Here is a closer image of a duct. The larger ducts have more rectangular columnar epithelium, whereas ducts within acinar lobules contain shorter, more cuboidal epithelium. This interlobular duct epithelium is a transition between the columnar and cuboidal epithelium. Here is an example of a larger duct. The epithelium is more columnar than cuboidal. Here is an intercalated duct. As an intralobular pancreatic duct, intercalated ducts are found among the acinar cells rather than being surrounded by connective tissue. The smallest of the pancreatic ducts, intercalated ducts, have cuboidal epithelium. In review, pancreas histology may be divided into acini, islets, and ducts. Acinar cells make up the majority of the pancreatic lobules and are densely packed with organelles and basal nuclei. There are 4 types of islet cells, but they are essentially indistinguishable through H&E staining. The pancreatic ducts are lined by epithelium which varies from cuboidal to columnar. This concludes our review of normal pancreatic histology.
Gallbladder, let's talk about the gallbladder. Gallbladder पित्ता कहलाता है और पित्ते में पथरियाँ होती हैं. आप लोग जानते हैं पित्ते की इन्फ्लेमेशन और इन्फेक्शन को कोलिसिस्टाइटिस कहते हैं और पित्ते में पथरियाँ हों तो उसे कोलीलिथाइसिस कहते हैं. It is a sac-like structure which is. As regards the structure of the gallbladder, let's talk about its histology. There are 3 layers: the tunics or the layers, the mucosa, muscularis and adventitia. There is no submucosa. The mucosa is thrown into numerous folds that are particularly prominent in the empty bladder. You can see this is the microscopic picture of the gallbladder. When we see under the microscope, mucosa is thrown into numerous folds. The lining epithelium of the gallbladder is simple columnar. It consists of tall cells with oval nuclei which are located in the basal region of the cell. The free surface of the cells bears short microvilli which are responsible for the presence of a thin striated border over the epithelium. Under the epithelium is present is a thin lamina propria consisting of delicate reticular connective tissue. The lamina propria is devoid of glands. But in the neck region, we have got the glands. The lining epithelium of the gallbladder is simple columnar. It consists of tall cells with oval nuclei which are located in the basal region of the cell. The free surface of the cells bears short microvilli which are responsible for the presence of a thin striated border over the epithelium. Under the epithelium is present is a thin lamina propria consisting of delicate reticular connective tissue. The lamina propria is devoid of glands. But in the neck region, we have got the glands. The muscularis of the gallbladder consists of a meshwork of interlacing bundles of smooth muscle fibers, between which are present collagenous and reticular fibers. Outer to the muscularis is a thick layer of connective tissue rich in elastic fibers. This is sometimes referred to as the perimuscular layer or connective tissue. The superior surface of the gallbladder is covered by an adventitia of dense fibrous connective tissue that binds the organ to the capsule of the liver. Elsewhere, the gallbladder is covered by the peritoneum, that is the serosa. Gallbladder, the functions of the gallbladder are the storage of bile, concentration of bile, acidification of bile, and it sends bile to the duodenum in response to cholecystokinin secreted by the enteroendocrine cells in the small intestine.
Here is one of the most beautiful gallbladder sections you'll ever see. The gallbladder is notoriously simple and has the classic appearance of a perfectly well-oriented epithelium of the mucosa, a looser lamina propria between that epithelium, which is sometimes a little bit papillary, and then the main wall of the gallbladder consisting of smooth muscle fibers to enable it to contract the way it likes to contract, and then looser connective tissue of the outside, which is chiefly going to be peritoneum rather than adventitia because it is mostly intraperitoneal. Let's take a quicker look. Let's look at these beautiful, 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 well-oriented, perfectly lined up cells of simple columnar epithelium. Look at the lamina propria in between. Look how the nuclei are arranged nicely at the base. Notice then you are very soon entering the general wall or smooth muscle of the gallbladder. Whether there's a true submucosa or not really doesn't matter, but you do know you have sort of a looser connective tissue between the epithelium, very soon developing into distinct spindly smooth muscle fibers of the wall itself. Then you can see the connective tissue of the serosa. Here's a large vein. Here's a medium or small artery. This looks like a vein to me. This is an artery. This is a nice branch of the artery. Here's a nice vein. Here's some fat and looser connective tissue. That may very well be a little nerve. And also at the edge of the loose connective tissue, you're very likely to see spindly little cells, which I can convince you are mesothelial cells, because we are truly now in the visceral peritoneum, or the serosa of the gallbladder. And want to go back in. Serosa, blood vessels of the serosa, muscular wall, lamina propria between the papillary structures of the mucosa, and here is the epithelium of the mucosa itself. And classically, when people want to give an example of a beautiful columnar epithelium, simple columnar, they always use the gallbladder because it always lines up very, very nicely, cell after cell after cell after cell, showing that it's a columnar geometric arrangement with the nucleus at the base. Thank you very much.